All right, welcome back. This is 10.2. Last section we went into kind of an overview of the process. Now we're going to go quite a bit deeper and start looking at the individual processes and how they work and how much ATP is produced in each one. So in glycolysis, one molecule of glucose is converted into 2 molecules of pyruvic acid. So glucose is a 6-carbon molecule. You can see that here with these 6 carbons. Pyruvic acid is a 3-carbon molecule. Okay, you can see there are 2 of them. So here's what that looks like. So glucose, this happens in the cytoplasm. ATP is brought into ATP, and that is what's used to break apart the glucose into the 2-pyruvic acid. So here we have 2 ADP being produced, and then we have NAD plus coming in to be charged, which turns into NADH. These are those electron carriers. And then we have ADP coming in to be charged up to ATP. So as a byproduct of this process, we have 2 net ATP, right? So we're using 2 and we're gaining 4. So our net is 2. We have 2 NADH and we have 2 pyruvic acid. Okay. So once the pyruvic acid is broken down into carbon dioxide, it goes to the Krebs cycle. And the Krebs cycle is just a series of energy extracting reactions. I'm not going to make you memorize every single step of it, but know that when you go to college and you take biology there, it's very likely that they will. So it's probably a good idea to have a basic overview of it now. So here's where the Krebs cycle happens. Hopefully you recognize this organelle, right? I said that glycolysis happened in the cytoplasm. Well, the rest of this happens in this organelle here. It is the mitochondria. So this process is why mitochondria is known as the powerhouse of the cell. Everyone always says that, but no one can really ever answer why is the mitochondria the powerhouse of the cell. It's because respiration happens there, right? Which is how we actually get our energy to perform cellular actions. All right, so let's go here. So pyruvic acid is broken down to carbon dioxide. Let's see a blow up of the actual process. So pyruvic acid from glycolysis is used to make NADH, ATP, FADH2 with carbon dioxide as a byproduct. So you can see here, okay, these are those NADHs, okay, here's the ATP. Let me zoom in a little bit here so you can see NADH, NADH, ATP. Here's that FADH2, okay, and then carbon dioxide coming off is right here, and right there. Okay, so the CO2 here is represented by the single carbon molecule with the 2 oxygens bound to it. So pyruvic acid passes through the 2 membranes of the mitochondria and into the matrix. So this is a little more detailed breakdown. There it goes. Once it comes through, the NAD plus comes in, it pulls off a hydrogen, which helps to break this down into this 2-carbon molecule and then CO2. And this 2-carbon molecule is called acetic acid. So we started with glucose, we broke glucose into pyruvic acid. Now we're converting pyruvic acid into acetic acid. Now, once it's acetic acid, it can bind with an enzyme called coenzyme A, which is right here with CoA, right? Which makes a molecule called acetyl-CoA. Okay, so here's this NADH being produced. We have acetyl-CoA coming out of this step. We have a CO2. Okay, let's move on to the next part of the Krebs cycle. So acetyl-CoA combines with 4 carbon compounds in the Krebs cycle, and that produces citric acid. Now, how many carbons do we have here? So you have a 6-carbon molecule here, so that makes sense, right? We have 4 coming in, we're adding the 2 from the acetyl-CoA. That gives us a 6-carbon molecule. So next step, citric acid is broken down into a 4-carbon molecule, which is going to release more CO2. Okay, so here's the process of breaking it down into a 4-carbon molecule. So the first break is going to release a CO2. Second break is going to release another CO2. Okay, and the energy is going to be used to create NADH. Once we have this 4-carbon molecule, the last step is to use some of this extra energy or these extra hydrogens to create FADH2 and NADH, and that leaves us with our original 4-carbon molecule, which can then pick up another acetyl-CoA and create citric acid and go through these steps again. Okay, what you need to know for this step, the Krebs cycle, glucose comes in, it's broken down. In the process of breaking it down, we're producing NADH, FADH2, some ATP, and carbon dioxide. Sitting through this. All right, so for each turn of the cycle, so each time we go through this cycle, okay, a molecule of ADP is converted into a molecule of ATP. Now, how many turns can this do per molecule of glucose? Well, we know one molecule of glucose is broken into 2 molecules of pyruvic acid, right? So that would mean 2 ATP would be produced from 1 glucose. Okay, there it is. So everything that's happening here is going to happen twice per molecule of glucose. So at 5 places in the cycle, electron carriers accept a pair of high energy electrons, and NAD plus and FAD are converted into NADH and FADH2. Okay. So the products of the Krebs cycle and glycolysis feed to the last step of cellular respiration. So far when we look at energy, how much ATP was produced in glycolysis? It was 2. Krebs cycle, it was 2. So far we have 4 ATP that we have netted, which is not a lot. Okay, this is a very, very small amount. The electron transport chain is the 3rd stage of cellular respiration and arguably the most important one. So the electron transport chain uses high energy electrons from glycolysis in the Krebs cycle to create ATP from ADP, or to synthesize ATP from ADP. So if you remember all those NADHs and FADHs, right, those were high energy electron carriers. Those come into play here. So let's go into it a little bit deeper. So the electron transport chain is comprised of a series of electron carriers located in the inner membrane of the mitochondrion. So if you look here, that is what we're looking at. So if you think back to photosynthesis, right, energy was used to pump hydrogen ions through a membrane to create a concentration gradient. And a very similar thing is happening here. So in this inner membrane space, we're going to have a lot of hydrogen ions, okay? And that's because we're using this energy to pump them through. So you can kind of see where that's happening here. Now, the ADP are going to come in, and they're going to link up with ATP synthase, right? So this is a process known as chemiosmosis, okay? So there's a higher concentration over here than there is over here. So what that's going to do is it's going to force the hydrogens to try and force their way through this ATP synthase. And as it does, it's going to cause the molecule to, or the protein to literally spin. And as it spins, it's going to generate the energy to bind this ADP with a free-floating phosphate group and create ATP. So on average, each pair of high energy electrons that moves down the full length of the electron transport chain provides enough energy to produce 3 molecules of ATP. So what is our net? Glycolysis, the Krebs cycle, and electron transport chain release about 36 molecules of ATP per molecule of glucose. So we have 2 in glycolysis, we have 2 in the Krebs cycle, and approximately 32 in electron transport chain. that number varies. So you'll see a variety of numbers, but the average is about 32. So that's the number we're going to go with. So your total ATP is going to be generally in the range of 30 to 42. In this case, we're looking at 30, 2, 4, 6. Okay, so 36, which falls right in that range. And we also have CO2 being produced as well. All right, and that brings us to our last section, which is for fermentation. Go ahead and answer the question, and then I will join you back for this video.